Alright, this is unit 3.3 and we're learning about the endocrine system. First, we're going to learn about the four types of hormones in the human body. So you have one, amino acid hormones. And following the amino acid hormone, you have a tip here. What does amino acid make up? Well, they make up peptides. So the second one we have is a peptide hormone. Now, what's the molecule that's larger than peptide? That's the protein. So we're gonna have protein next. Peptides make up protein. So our third hormone is a protein hormone. And the fourth is kind of different. It's not big protein hormone. We have for the fourth one steroid. And I like personally sort these into two categories. The AA peptide and protein one category and the steroid in its own category. The way I sort them into two categories is because these three are kind of all related, right? Amino acids make up peptides, and peptides make up protein, and you can see it's just an easy way to remember them all. Now, a second reason I sort them into these two categories is because they fundamentally work different. So, in the example of a peptide or protein hormone, let's say you have the cell membrane here, and inside the cell membrane, you have the nucleus and some DNA in the middle. For a peptide or a protein hormone, so coming like this, it would have a receptor on the membrane. And the active side of this receptor is complementary to the 3D shape of the hormone. So the hormone coming fit and bind with the receptor forms a complex. And this binding causes a new molecule. So that's another molecule inside the cell to go and enter the actual nucleus to initiate change, to bind with the DNA and initiate transcription. So that's how peptide and protein hormone initiates change in a cell. But a steroid hormone works very different. Same setup, you have the cell membrane and the nucleus, some DNA inside. But this time you don't have the receptor protein on the cell membrane because a steroid hormone it's a lot smaller. It doesn't need a receptor protein because it just kind of diffuses into the cell membrane. So it just comes in here, and initiates a change to the nucleus just by entering the cell. All right, next thing. The next thing we're gonna learn is glucose regulation, the regulation of glucose level in the bloodstream. And there are two major hormones that regulate this process. And we draw this as a negative feedback diagram. So in the middle, you have a horizontal line. You draw the line first, and you draw an arrow going up and an arrow going down the other side, and vice versa on the bottom half of the diagram. So what this line represents is like the threshold, the normal glucose level. When it's going above this line, that means there is too much sugar, too much glucose in the blood. When it's below this line, it means you're not having enough glucose level. So what this means is what this line represents could be someone's blood glucose level after he eats like 50 chocolates. It's skyrocketing. It's really high, right? It's getting unhealthy. And if this person has a healthy, normal body, doesn't have diabetes, and can just function well, his body would do three things. Actually, his body would release insulin first. Insulin. And that's the thing you see diabetic people, like before they have a meal, that's the thing they inject into their body. So insulin lowers the blood glucose level when it's too high. And the way it does this is by initiating or uh, inhibiting these three reactions. So the first one you need to know is glycogenesis. 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 So this is the making of a glycogen from glucose. Now this is another biology knowledge. Our body can have glycogen. It's a form of complex sugar. It's stored in the liver and muscles. Our body is fine with dealing with that. But when there is a lot of glucose in our blood, our body doesn't want that. So to reduce the amount of glucose, the body converts the glucose to glycogen, which is the body is fine to deal with. So what happens when there's too much sugar? Glycogenesis is promoted. So this process happens. Thus it gets a tick. The opposite of glycogenesis is called glycogenolysis. And this is the breaking up of the glycogen into glucose. And you don't want that. You don't want this when there is too much glucose already in your bloodstream. And the third one is this is the one with confusing name. This one is called gluconeogenesis. Gluconeogenesis. So this is the process of converting proteins and fats into glucose. Again, you don't want more glucose in your blood if you already have too much glucose. Now, these are the three actions uh, that insulin does. Where's my mouse? All right, found it. Um, okay. So on the other hand, you have the glucagon. Glucagon. This is the hormone that your body releases when you have not enough sugar, right? When your body needs sugar, it releases glucagon. And it does the exact opposite uh, uh, functions that insulin does. So let's assume we have glycogenesis up here, glycogenolysis, and gluconeogenesis. It would inhibit glycogenesis. It would promote glycogenolysis and it would promote gluconeogenesis. All right. Now,